Welcome everyone, it's Dr. Lindner. In this video, we're going to be looking at the neuromuscular junction. We're going to focus on a little bit of the physiology of what takes place at the neuromuscular junction, and then we'll show you the model of the sarcomere. So when we talk about the neuromuscular junction, sometimes we abbreviate that as the NMJ, neuromuscular junction. Well, here's the muscle, here's a muscle fiber, here's a somatic motor neuron, right? Here are the dendrites or the telodendria, depending on how your instructor teaches you that. Dendrites or telodendria. So these dendrites bring information into the cell body or the soma, and then it sends out information into this axon. And right around here, they refer to this dilated portion as the axon hillock, and then here's the axon all the way down, and you could see that there's this insulation. Each of these little rolls, it looks like, or hot dog buns wrapping all the way around. These are Schwann cells. Schwann cells. And Schwann cells are a type of neuroglial cell. It's part of the neural system. When we start covering the neural system, we'll discuss two types of cells that make up the neural system. One is a neuron, which is what you see right here. This is a neuron. And the other are called neuroglial cells or neuroglial cells. And they are considered to be supporting cells of the neural system. So here we have these Schwann cells that serve the purpose of wrapping around and around and around and around the axon to insulate and protect all this delicate wiring. Because really that's what I think of when I think of neurons and nerves. I think of electrical wires that need to be protected. Because if you touch an electrical wire with your finger, you'll get electrocuted, you'll get shocked. So we want to make sure that these axons are insulated and protected. Right? If you look at anything that you plug in, even like a smartphone charger, a toaster, a TV, you always have to plug it into a source, but the plug is insulated. Usually it's the black coating around the wires. It's insulated. And if that wire ever breaks down and you see, if that black insulation ever breaks down and you start to see the wires inside, you have to be real careful not to touch them. So here we have this insulation that's made up of cholesterol and fat and sphingolipids and phospholipids. All of this is like a fatty type of cholesterol that acts as an insulation to protect and to speed up the nerve conduction from here to here, to really speed them up. Now, there are conditions that we call autoimmune conditions, where the body starts attacking itself in autoimmunity. There's an autoimmune disease where the body can start breaking down its own myelin. That's what this is called, myelin, that insulation. Myelin or myelin sheath. Myelin or myelin sheath. Okay. That's what this insulation, this fatty cholesterol is. And if the body has an autoimmune condition where it starts to break down the myelin, well, that's what we call MS or multiple sclerosis. Okay. So we have these Schwann cells that insulate and protect the axon. And at the very, very end of the axon, we get to the axon terminals. We call these terminals at the very, very end. And at the very end of the terminal, right in here, we have what's called a synaptic a synaptic vessel or synaptic vesicle. Synaptic vesicle. And at the very end of the synaptic vesicle, that's where this terminal part of the axon makes contact with the muscle, which is really the neuromuscular junction. So something is happening at that synapse. So right here, this part would be the end of the axon, where the axon terminal is, where we have synaptic bulbs that carry synaptic vesicles. And on the other side here, we have the motor end plate. This is the muscle portion. All of this is the muscle. So we have the neuromuscular junction. This is the very, very end of the nerve where we have the axon terminal. And at the end of an axon terminal, if here's the axon, there's going to be this bulb at the end, the synaptic end bulb. And then here would be the motor end plate of the muscle. And inside of the bulb are going to be these little vesicles that we see right here called synaptic vesicles. And these synaptic vesicles store or house neurotransmitters. Neurotransmitters are just communicators, they're messengers. Well, the type of neurotransmitter that we find here is called acetylcholine, ACH, acetylcholine. Now, these vesicles have to move towards the end of the end bulb. They move in this direction, and they're going to open up, and they're going to release the contents. They're going to open up and release this neurotransmitter into this synaptic cleft, this gap or this space between the neuron and the muscle. What's going to promote this synaptic vessel to open up and release its contents? And if you think back, there's a special term for that. There's a special term that we use to express that a vesicle is opening up and releasing its contents outside. If you remember, we call it exocytosis. Exocytosis. That's when this vesicle opens up and releases its contents into the synaptic cleft. Exocytosis. And what's going to promote that, what's going to tell it to do that, is the influx of a mineral called calcium. So calcium is going to rush in here, and it's going to tell the synaptic vesicle to release the goods, to release the acetylcholine. And acetylcholine is always, always, always excitatory. It's always excitatory. I put a plus sign there for excitatory. Acetylcholine is always excitatory at the neuromuscular junction. Now, the acetylcholine has to find some place to hook up with, and it does. The acetylcholine that is released has to meet up with these acetylcholine receptors. These are acetylcholine receptors. So when this meets up here with these acetylcholine receptors, it's going to open up and it's going to allow something to enter the muscle cell. And that something that's going to enter the muscle cell is what we call sodium. So sodium rushes in. And when sodium rushes in, it creates excitation or depolarization. Excitation or depolarization. And when you don't need that excitation or muscle contraction anymore, the acetylcholine has to be broken down. So we have these things here that look like these crescent-shaped things that are going to eat up the acetylcholine, and that's called acetylcholine esterase. There's an E for esterase, acetylcholine esterase. And remember, anything that ends in ACE is an enzyme. Okay, so we have the acetylcholine through exocytosis that's in the synaptic cleft. It has to bind to these channels, these receptors. When it does, they open up, sodium rushes in, and it creates excitation down this invagination right here. These invaginations or indentations are called T-tubules. T-tubules. And that's going to be important. You're going to see that in just a minute. T-tubules. So now let's take a look at the muscle fiber and let's see where some of this comes into play. So this portion here, this is all going to be the muscular side and all the different things that happen in a muscle. And over here, this is where the nerve is going to come into contact. All this yellow is going to be the nerve that's going to meet the muscle. So we're looking at the neuromuscular junction. Okay. On the outside portion, we have this layer of protection called an endomysium. Endomysium. And this is a Schwann cell here. I'll show you another picture of what that looks like. So endomysium, what you're going to learn in lecture is that we have these muscle cells like this. These are these little muscle cells here. And these muscle cells are going to have a layer of protection around it called an endomysium, right? Each of these will have an endomysium going around it like this. Okay, each of these muscle fibers are going to have this endomysium going around it. And then I can cluster these like that, and I can cluster these, okay? So that's called a perimysium. That layer is called a perimysium. And if I group these together as a group, that's an epimysium. So you have an endo on the inside, a perimysium, and then going around the whole thing, an epimysium. Okay, when we look at this, we are going to see a few different structures. Let's focus on, here's the endomysium that I pointed out. It's kind of like that membrane going around the entire sarcomere here. Muscles have lots of mitochondria. You see mitochondria here. This membrane right here is called a sarcolemma. Sarcolemma. This type of fluid structure here that we see deep to these folds, that's called a sarcoplasm. Sarcoplasm. So here's the sarcolemma. It's like the membrane that's just deep to the endomysium. And then here on the muscle side, where you have the mitochondria and you have lymph and you have arteries and veins, that's going to be your sarcoplasm. Now, when we spoke about action potentials a little bit earlier, action potentials are going to drive down the T-tubule. Remember here, we spoke about the T-tubule. When sodium rushes in, it creates excitation. It goes down the T-tubule. So right here, this blue structure is the T-tubule. It stands for transverse tubule. Why? Because it runs transversely to everything else that's kind of going this way. Okay, so the electrical conduction is going to travel down the T-tubule, and this tan spider web mesh-like webbing is called a sarcoplasmic reticulum. Extremely, extremely important, because what it does is it houses a really important mineral called calcium. So all in here is where calcium is stored. And calcium is somehow involved with muscle contraction. When the action potential moves down and gets transferred into the sarcoplasmic reticulum on both sides, right? You'll see that the sarcoplasmic reticulum surrounding each transverse tubule. We can see it here. So when the sarcoplasmic reticulum releases this calcium, calcium is going to have a major impact on these muscle fibers, where we have these contractile proteins called actin and myosin. Actin and myosin. And somehow myosin kind of looks like it has a tail and then it has a head, has to connect with actin, which is like a bunch of these little pearls or circles. And within actin, there is this blue circle in each of these. These are called binding sites. Okay. And if these binding sites are open and exposed, these myosin heads connect to these blue dots, to these binding sites. They connect and they make a contraction, the way they interact with one another. But what's really neat is that these binding sites are actually covered by a protein. So they end up being covered by these green, in this picture, I just use green, these green ropes or strings that are covering the binding sites, and those are called tropomyosin. Tropomyosin. So tropomyosin is another type of protein that's blocking those binding sites. And then there's another protein that holds it in place. It acts as the glue. We'll use black. So there are these black staples that hold them in place. And those black staples are called troponin. Troponin holds the tropomyosin in place. It's the staples that hold the strings in place. Now, when this calcium is released, when the calcium is released and it's sprinkled down onto all these muscle proteins, the actin and myosin, what calcium does is calcium goes after the troponin. So it goes after these staples, and calcium is like the staple remover. It removes the black staples, it plucks them out. And if the staples are gone, that means tropomyosin is free to move. It can actually shift out of the way. And if it shifts out of the way, those blue dots or the binding sites are now exposed for myosin to connect to. So again, we have these myosin heads. We have actin. And actin has these black dots, these binding sites. And when they're exposed, the myosin head magnetically can connect to them. And when they connect, they create a contraction. But we have this tropomyosin, which is like another protein that's covering the binding sites, and they're held in place by troponin. Calcium comes in, removes troponin, so tropomyosin shifts out of the way, and the binding sites are exposed, and a muscle contraction can take place. So you may want to, when we're done, pause and then go back and replay this just so you can kind of hear it again. But the muscle physiology is very complex, but that right there is it in its simplicity. It really is. So you should know the transverse tubules. You should know the sarcoplasmic reticulum. You should know that the sarcoplasmic reticulum is there to store calcium. And where the T-tubule and both sarcoplasmic reticulums connect with it, they call it a triad because it's really three structures. 1, 2, 3. So they call it a triad. Okay. When we take the muscle and we look at it from the side, like bird's eye view looking down, these are the myofibrils. Everything that you see here, these are the myofibrils. Now, remember, each of these has like an endomysium around it. Okay. Each of those has an endomysium around it. So here, these are myofibrils containing actin and myosin. Actin is thin, myosin is the thicker filaments. So actin is thin, myosin is thicker. So here, actin is thin, myosin is thicker. And it's these thin and thick filaments, when light passes through this on a microscope, it's what gives it its striated appearance, right? Because skeletal muscle is striated. Okay. I'm not going on lecture. They'll probably ask, you know, what is the A band or what is the H band or the I band? I'm not going to hold you accountable for that. These bands are really more important if you're studying exercise physiology or physiotherapy or physical therapy or chiropractic. That's where these things get a little bit more into play. But this entire unit between actin and myosin, the way all this interacts, we call it a sarcomere. That's called a sarcomere. The sarcomere is the functional unit of muscle. It is the structural and functional unit of skeletal muscle. Let's take a look at the axon. So here's the axon, and the axon is insulated by this Schwann cell, right? That's what's going to insulate and protect this, and it just really wraps around, and wraps around, almost like a jelly roll, to create all these layers of insulation. But this is one Schwann cell. This is the nucleus of the Schwann cell. And even nerves have mitochondria. So this is just the mitochondria that are found in the axons. If I'm going to ask a Schwann cell or the nucleus of the Schwann cell, I won't ask it here. If I'm going to ask those, I like to ask it in a more obvious view, which is this view here. Okay. What we can see is that the axon comes down, and you can see all these little vesicles. Well, if you remember, these little small circles, those are the synaptic vesicles that contain acetylcholine. So in those vesicles, we have ACH, acetylcholine. This is all nerve. This is all muscle, right? So this is where the neuromuscular junction is, right here. Let's take a look, see if we can close that, take a closer view. So here's the Schwann cell. Here's our axon. These are mitochondria. These are the synaptic vesicles. What's in those synaptic vesicles are acetylcholine. Acetylcholine. So now here is where the nerve side comes to an end, right? This is where the nerve side comes to an end. And right here, this is where the muscle portion begins. So they're making contact with one another. So this is the nerve, this is the muscle, this is the neuromuscular junction. So they call this the pre-synaptic membrane, and this is the postsynaptic. Pre is nerve, post is the actual muscle. And in between, this is the synaptic cleft. The synaptic cleft is the gap or the space between. Okay, let's take a break here. See if there's any questions.